So we're going to talk about DNA sequencing technologies. To start this off, I want you to imagine that you're deep undersea. You're exploring, you're swimming around, and you see this really bright pink organism, something you've never quite seen before, swimming around. It's got lots of tentacles, maybe like a little bit of a cross between an octopus and a jellyfish, but it's glowing, this glow that you've never seen before. And so you reach out towards it and you touch it. But as you touch it, it gets scared and it drops one of its tentacles and swims away quickly. Sort of the same way that a lizard might drop its tail when you grab it. It knows that if it releases whatever is, got your attention, then maybe it can get away faster. So you're left there in the dark now with just this tentacle. It's still glowing a little bit, and you're intrigued. You want to know more about this organism. You want to know whether that glow could be useful for something else. So my question to you is, what can you do with that tentacle to find the source of the pink glow? Is there any information in that tentacle that you can get at in order to figure out how this deep sea creature was glowing and whether or not you can engineer that same feature into other organisms? Well, if you had its genomic sequence, you could predict the genes that are encoded within its genome. Here you're just seeing a zoom in on a piece of DNA, and each protein encoded in the genome will start with a start codon. Something like ATG is the most common 3 letters that encode for a start codon, although occasionally a TTG or GTG will also be able to serve this function. And then in frame, that means a multiple of 3 bases downstream of this ATG will be a stop codon. There are several stop codons. TAA, this one that you're seeing here, is one of them. And these are the codons that tell the ribosome, you've reached the end of a protein, you can let go now. So if you find a start codon and stop codon that are a multiple of 3 bases away from each other, then you've likely found a gene. So in some ways, just looking at genome sequence can allow you to predict where genes are encoded within the genome. Now, we talked a little bit about splicing in the previous lecture, and so you'll know it's not quite this simple, and there are mathematical models, there are ways of trying to predict gene sequence, even genes that have introns in them that may shift the coding range or the coding window. But at a high level, this is something that's possible to do. It's possible to predict where genes might be encoded in the genome. And it's becoming increasingly possible to also predict the function of those genes. Before we knew a lot about proteins, we didn't really have an idea or a way of predicting what an individual protein might be doing. We would just know that it was possible that that region of the genome encoded for a protein. Now we have the really nice data from a lot of organisms that we can compare our genes to. So we can do something like a protein sequence alignment, where we take a protein from our mysterious deep sea creature and we align it. We look for matches in the human genome, in the chimp genome, in a mouse, or in another well-studied organism. And we see, are there any regions of those genomes that look like the region of the genome from my deep sea creature? That might tell us if there's a high match in this case, the protein sequence. That might tell us that the proteins serve a similar function. We also talked before about how structure can be more important than the sequence of the protein, as it's actually the 3-dimensional structure of the protein that is contributing the most towards the function. And so using something like AlphaFold, you might be able to predict the structure of a gene encoded in your deep sea creature's genome, and then align the structure. See, these are 2 different proteins doing a structural alignment. One is in red and one is in yellow, and you can see that their structures are very similar. So by doing something like this, you can have an even better idea that maybe these 2 proteins serve the same function. So with this capacity to predict what genes are in a genome and what those genes may be doing, what the proteins that they encode for may do, Then it becomes really exciting to start applying DNA sequencing technologies to all kinds of organisms to start to try to figure out what are they doing, what are they capable of doing, and how are they doing it. I do want to point out that our knowledge of gene function is still pretty limited. And so it's likely that there will be large portions, most of the genome of this organism we wouldn't necessarily understand. But we have some tools to begin to figure out what portions of a new organism's genome might be doing. Now, how do you figure out the sequence of this organism's genome? Well, if we're 1980, if it were, 40 years ago, you might try DNA sequencing with the Sanger method. Sanger is essentially DNA sequencing by synthesis. This is a picture of Fred Sanger. He invented this method. And just want you to remember that usually when we synthesize DNA, we have a template strand. And then we have the strand that you're building. And so you'll incorporate individual DNA bases to pair with the template strand. And so Sanger invented a method of observing what bases are getting incorporated as DNA synthesis is occurring. And by reading out which bases are getting incorporated, you would know the sequence of the template strand. Now, the thing that so usually DNA synthesis requires deoxynucleotides. So here's the sugar backbone, the base, that A, T, C, or G is just indicated by this little circle that says base here, but just imagine that there are nucleosides here. And then the sugar backbone has an OH group at one end and a triphosphate, that's what these things are, at the other end. And this oxygen or OH group interacts with the triphosphate to catalyze a reaction that leads to the attachment of this new nucleotide onto the growing DNA strand, right? So this presence of this OH group is really important for the elongation of a DNA molecule. And this is what's happening in all of your cells as your DNA replicates. This type of chemistry is occurring. But what Sanger found was that if you use a dideoxynucleotide, so something where there are no OH groups on the base, then it stops DNA synthesis. You essentially terminate the reaction. You can no longer elongate the DNA. Now, why would that be useful? Well, if you make a mixture of 4 different solutions and you try to run PCR, polymerase chain reaction, you try to synthesize the opposite strand of a DNA piece that you want to sequence, and you have a mixture of dideoxynucleotides and regular nucleotides in each reaction condition, then you'll get DNA synthesis that stops at different points. So if you're synthesizing, you always synthesize from the 5' to 3' direction. So this might be the first base that you try to synthesize. And if you're in this if you're in this reaction condition, then you'll probably incorporate a DDTP, a dideoxynucleotide, and stop the growing reaction. Whereas if you're in this DDCTP, you may elongate with that one base and then have a stop at C. But what I want to point out is that this contains a mixture of nucleotides that are both dideoxy and deoxynucleotides. So occasionally in even this reaction condition, you'll incorporate a T as normal in this position and then go on to hit another T later where you incorporate a terminal nucleotide. Okay, why does this matter? Well, that means that in this reaction, you have a collection, in each reaction actually, you have a collection of DNA of different lengths. And then you can run those out on a gel using gel electrophoresis to see how long the base or the segments of DNA are. So in this case, if we had this T is supposed to indicate a reaction in which you had all 4 regular nucleotides and then 1 dideoxynucleotide TTP. And that one, you had one really short fragment corresponding to this one base long DNA sequence, and then one kind of medium fragment corresponding to here. The A, you had a different length of molecules because it was terminating every time it reached an A. Here you're terminating every time you reach a G, and here you're terminating every time you reach a C. So if you want to go back and read the sequence, you would say, okay, the first base is a T, the next base is a C, the next base is a G, the next base is an A, right? And you can do that because you know that the sequence of this length is only possible because we incorporated a stopping nucleotide at that location. Okay, so hopefully that's clear. I thought it was it is a very clever way of trying to read back DNA sequence because it's actually not that easy to do. And here's an example of what one of these Sanger sequencing gels actually look like. You can see how, each length of DNA leads to a slightly different, each reaction leads to a slightly different collection of DNA lengths, and you can read back out what the sequence might be. But notice, you probably have 20, 25 bases read here. And if the human genome is 3.2 billion bases long, it would take a very long time to use Sanger sequencing gels in order to sequence the entire thing. It's not to say you couldn't do it, it just might take quite a while. So people started doing things that would not require you to run a gel at the end, since reading back out that gel is quite time consuming. Actually developed these dideoxynucleotides. Remember, these are the ones that terminate the chain reaction, the elongation of DNA, that have fluorescent dyes attached to them. And so as you synthesize DNA along your template, if you incorporate one of these chain terminating fluorescently labeled nucleotides, then you would end up with a piece of DNA of a specific length with a specific color dye at the end. And then you can take this big mixture. You don't have to do 4 different reactions. You can do them all together since each nucleotide has a different color. And you can run them through what's called a capillary gel. So now instead of having a big gel where you're able to visualize each lane, it's this very thin tube that's full of gel, and it still separates the DNA based on size. So the smallest pieces of DNA will run through the fastest. But as you are running the DNA through, you're actually using a laser to excite these fluorophore dyes, and then detecting to see, okay, well, if did I see a red dye? Did I see a green dye? Did I see a blue dye? And based on the order of the dyes coming through, what the sequence is. So this type of reaction gives you data that looks like this. We call these chromatograms, where each peak corresponds to the passage of a DNA of a specific length through this laser detector region, showing you that there's a piece of DNA that's this length that has this specific base. In this case, I think pink is a G incorporated in your DNA. So this has helped accelerate DNA sequencing quite a lot. Up until the last year or two, this was the most common method used by my lab, and that I used all throughout graduate school to do DNA sequencing. But there are some newer, what's called next generation sequencing technologies that will allow you to do even more sequencing, to get at even the sequences of even bigger genomes. Because again, these types of reactions, they're very good up until about 1,000 bases of DNA, but it would still take a really long time to sequence, a human genome if you're doing it in 1,000 base pair chunks. Okay, so there are these next generation sequencing methods, and these are what we're going to talk about today. I think that they're all super interesting. It's really cool to see how people have figured out how to sequence DNA. We're going to go through kind of how they each work and what the benefits and drawbacks of each method might be. So the first one that we're going to talk about is actually a variation on sequencing by synthesis. We're calling it next generation sequencing by synthesis, and this is what is used by a company called Illumina. The machines that they build actually look just like this. Okay, so Illumina sequencing is sort of similar in that you're going to be incorporating bases with a mixture of either dyes or, you're going to be incorporating a mixture of bases, each of which has a dye on them. And these are Yeah, all done on a chip instead of in solution. So what happens is you take a bunch of genomic DNA and you shear it into pieces. You can actually agitate a solution of DNA in a way that breaks it into fairly regular pieces. Then what you do is you ligate on, you attach little short DNA sequences called adapters. These adapters all have exactly the same sequence. And what they're going to do is allow you to bind your pieces of genomic DNA to what's called a flow cell. So this is a little chip that basically has tiny pieces of DNA already stuck onto it. And those tiny pieces of DNA are complementary to your adapter sequences of DNA. So those little adapters allow you to stick pieces of genomic DNA onto the chip. And then you do something called bridge PCR, where what you're really trying to do is get clusters of DNA that's all identical right next to each other. So it's really difficult with this Illumina method to sequence a single molecule of DNA. So they do amplify it in this location. We can talk about how this kind of bridge amplification works if you're curious, but I think it's sufficient to just know that it occurs and it gives you clusters of DNA that's all identical in little locations along this chip. And then what you do is you flow through across this chip a solution of nucleotides that are each terminating, but they have a special chemistry. So they're terminating in that you can only incorporate one of these bases at a time. And each one of the bases are color coded. So you might have an A be red, a T be yellow, a C be green, and a G be blue. And you can then see, as you synthesize the strand of DNA, you can take a picture of the chip and see what color occurred at the location of each of your pieces of DNA. So what do I mean by that? Okay, so like if you can think of this as going in time as we move from top to bottom. So if the first base is red, then, or an A, then it's going to appear red. And then you'll wash out all of the remaining bases. You'll do what's called unblocking, where you then make the base ready to receive a second base. You'll flow in a new set of nucleotides, and a T might attach there, and so you'll read it as yellow. You'll repeat, and the next time it might be green, the next time it might be blue. And so as you image that spot over time, you'll see the color change, and that corresponds to the sequence of the DNA that's at that location on the chip. This is what one of these chips look like. So this is a flow cell. It's pretty small, 75 millimeters by 25 millimeters. This is what an image of one of those flow cells look like right after you've flowed your nucleotides over it. So you can see, the individual spots where there's DNA adhered to the chip, and the color indicates what base was incorporated then. There are about 1.6 billion of these spots per flow cell. So you can now measure like 1.6 billion molecules of DNA clusters of DNA simultaneously. So I think that's pretty cool. Now, this is what this looks like in real time as you are, well, it's maybe a little sped up, but this is what this looks like over time as you flow in and out different chemicals, different bases. You do that blocking, unblocking step. You have to track individual spots and see what color comes up. And so it's actually a big image processing problem to figure out how to track each of these, how to know, when the color has changed to indicate a new base has been incorporated, and all of that stuff. But I think it's pretty impressive. This type of sequencing method is very powerful. It can sequence a lot more DNA than the Sanger sequencing method we talked about before. Now, one of the problems with Illumina sequencing is that it generates short reads. So it only can read DNA sequences that are around 75 to 300 bases long. And that's okay, except 75 to 300 bases is not a lot of bases of DNA. And when you want to figure out how the whole genome is assembled together, then you would assemble each of these little short reads into contigs. So what does that mean? That means you would basically look for regions where they overlap slightly, or significantly even, and say, okay, well, clearly like these two segments of DNA are, they're next to each other, they're like partially overlapping. Because when you do that shearing, you have to remember that there are billions of molecules of identical DNA in there, and each one's going to get cut slightly differently. So you're going to have lots of overlapping sequencing reads. But anyway, these are assembled then into what's called scaffolds, where we fill in with ends. We basically say, like, we're not sure what sequence is in between these two larger segments of DNA, but we believe that there is missing sequence there, because sometimes regions get difficult to resolve, and we'll go into what that means on the next slide. Okay, so it can be challenging to sequence genomes with repeated sequences using this type of short read technology. When you're only getting sequencing reads that are 75 to 300 bases long, then you have a hard time figuring out whether like repeated sequences are real or not. So here's an example where you have a genome, let's say it's all mostly in orange, and all of the orange is unique sequence, right? That sequence that's only appearing once in the genome. But you have this repeat region where there's a segment, maybe several hundred base pairs long, that's identical to another segment in the genome. And in between them is some unique sequence. But we don't necessarily know whether these reads here map to this repeated region or this repeated region, because you'll notice like this blue bar, for example, it's completely contained within the repeat. So it's totally possible that it is a read from this region of the genome. And so when you use an algorithm to try to figure out how to assemble the genome, it becomes really difficult to know, are these reads two different repeated regions within the genome, or just reads covering one location? And these ones that are overlapping at the boundaries, maybe that's suggesting, this boundary here. So the algorithms have a hard time, people have a hard time figuring out how to deal with these repeated sequence, even though, there is this extra DNA in the middle, and there are some unique junctions here. When there's a lot of overlapping sequence, it still just becomes very difficult to know how to deal with that. And so some of that will end up as kind of trash at the end of an experiment, where people will say, well, these extra reads, we didn't really know where to align them. We didn't know where to put them. So, when you were going through the p-set and you see like the human genome project had finished, and yet we only had sequence of like 92% of the genome, some of it is because we just have regions that we didn't know how to kind of put into the context of the rest of the genome. Until recently, that has been figured out. So hopefully you'll enjoy seeing that on the p-set. Okay, our Illumina next generation sequencing has several advantages. It's cheap, it's very accurate, it's really good at accurately conveying what base is on a molecule. Those kind of clusters of identical DNA all being read at the same time means that it's pretty high fidelity. And it can sequence fragmented DNA. So if you have DNA that's old, right, and already pretty broken, or you have a sample that hasn't been treated very well, maybe it was, left in a hot car, or you were trekking from your deep sea adventure, you had to take that DNA on a plane and get it home. Things can happen that break down your DNA, and Illumina is still pretty good at sequencing even fragmented DNA, because it's going to break the DNA into pieces anyway at the beginning of the library prep. Some of the disadvantages of Illumina sequencing is that it's hard to sequence repetitive regions, and it can't resolve structural variants. So that becomes really difficult to do with this. Now, the next type of sequencing that we're going to talk about is single molecule real-time sequencing. This is different. So here you're taking sequence that you want to know the sequence of, and you ligate on these adapters. So you make a full DNA circle. And then at the bottom of a nanowell, a very small sample size, very small little like well in a chip, you have a single polymerase fused to the bottom. And that polymerase will incorporate or try to make a copy of this single strand of DNA. And as it does, it will incorporate fluorescent nucleotides. Then you can use basically an imaging tool at the bottom of this microwell. You can shine a laser and then collect the light that's re-emitted from the fluorescent nucleotides. And as they're getting incorporated into the DNA, they will be the closest to the detector and therefore give the brightest fluorescent signal. And so you can use that to figure out what the sequence of DNA is of this molecule as it's being synthesized. So this is essentially another sequencing by synthesis method. Here's what something looks like where you have fluorescence intensity over time, and you can see each base has a different signature. And so you would know that it's an A getting incorporated based on how intense the fluorescence signal is that you're reading at your detector over time. So it's true for each individual base and allows you to read DNA. One thing I will say is that this method is pretty low fidelity. So it's sometimes confusing to know whether it was an A or a T that got incorporated, a G or a C. It's kind of low fidelity, especially as you get fluorophores or some of these nucleotides that kind of drift down towards the detector. You can get errors in the sequencing read. And so what people usually do is run this circular DNA molecule through the polymerase over and over and over and over and over again, and then look for kind of the consensus sequence. If you read the same molecule many, many times, then you can kind of average out any error that's occurring. So that's helpful. Here's the actual size of one of these smart cells. So what's in there? Well, you have, right, this little micro well with the polymerase at the bottom. And then there are, and then it's placed into this chip. It's just kind of stepping through different magnifications here so you can see kind of what's happening inside of these smart cells. So it's an exciting method of being able to take long read measurements where you can take a long molecule of DNA and sequence the whole thing instead of having to break it into those small pieces like you do for Illumina sequencing. This PacBio, this SMRT sequencing can handle long molecules. And what that's good for is actually looking for repeated sequence. So now you can actually know how many times does a repeated sequence occur in the genome? Are there locations where a region of the genome is repeated or inverted? And sometimes those repeats or inversions can influence how strongly a gene is expressed. So they're really important. And this type of sequencing read has finally allowed us to be able to look at these types of kind of complex genomic regions. Okay, so it has these advantages of being able to sequence repetitive regions and resolve structural rearrangements, but it's not compatible with fragmented DNA. Again, the advantage here is being able to sequence a long segment. If you have a really short piece of DNA, it would like zip through the polymerase really fast, but not be a super good use of the technology. It does have a little bit of coverage bias, so it's not perfect at all reading all bases, and then it is low accuracy, although, again, we kind of get over that low accuracy problem by sequencing the same molecule many times in a row. All right, our last technology that we're going to talk about is nanopore sequencing. This is not sequencing by synthesis. This is a little bit different. You basically have a sensor chip with an array of nanopores, little holes on them, and you then measure the current as it passes through these nanopores. What does that mean? Okay, so on this chip, there's like I said, this array of tiny holes, these nanopores. They're embedded in a membrane that's connected to a chip. You can kind of see all of that here that measures current. And the current is disrupted by the passage of DNA bases through the pore. And so here's just an animation of this kind of thing occurring. In this case, it's sort of modeling the DNA bases of different kinds as these orange balls of different sizes. And you can see the current passing through on the left. So the current's passing through at a high level until that first base or that first ball occurs and it dips. The second ball, which blocks more of the current, gives a bigger drop. And, when it's removed, then it returns up to this baseline. And so each nucleotide interferes with the passage of ions through this pore differently and gives a unique signature. So you can sequence the DNA as it goes through. So something like that looks like this, where here's the DNA passing through the nanopore. And I apologize, that's super fast, but it's all passing through slowly, and you get this signature of different currents that correspond to the different bases passing through, and therefore the sequence of the DNA. Nanopore has all these great how it works videos. I totally recommend checking them out and seeing for yourself, what you think of these different technologies. And they do explain it in greater depth. So you can, please feel free to check those out. Okay, so Nanopore products are really cool. They're actually quite inexpensive, right? A MinION is about $1,000. So it's like a handheld version of a sequencer. And it includes 2 flow cells and a kit to help you kind of prep DNA. So for, around $1,000, you can sequence potentially 2 organisms' genomes, although they'd have to be relatively small organisms. And we'll talk about that in a second. But yeah, the prices here are they're pretty affordable relative to the cost of a lot of laboratory equipment. So it's been really exciting to have Nanopore on the scene because it's made it easier to sequence all kinds of DNA samples. Each product, again, has a different sequencing capacity. So the flow cell sizes, the number of nanopores on each one are different. And so the more nanopores you have, the more kind of chips you put in any one machine, the bigger the genome you can sequence. And so here's actually wheat has a genome that's even bigger than humans. Instead of being about 3.2 billion bases, wheat is around 16. So the bread that you eat actually has more DNA in it than you. But I think it's great to have this diversity of options available at different price points to be able to sequence DNA for all kinds of needs. Okay, this is something that I'm super excited about. I haven't seen it come out yet. We did talk about it last year and it's still not out, so I'm not sure exactly when it will happen. But Nanopore Tech, Oxford Nanopore, the company that makes these types of sequencing devices, has been talking about smidge ion. So if MinION is the version of the sequencer that you can plug into your USB port on a computer, smidge ion is one that you can plug into your cell phone and get some sequencing to occur. And I just think it would be so fun to be able to have a little device that you plug into your phone and that you can take sequence of maybe in the field or on the fly. So here are some of the advantages and disadvantages of Nanopore. It can sequence repetitive regions. It also does really long reads like the PacBio system that I showed you before. It's pretty cheap. It can resolve those structural rearrangements and it's portable, but it does have a lower accuracy than the Illumina system, and it doesn't have the advantage that PacBio does of being able to sequence multiple molecules over and over and over again. And so you are subject to potentially more mistakes in your sequencing. But if you have many copies of the same piece of DNA and they're all going through different pores, any one pore may have a problem getting a really accurate sequence read. But when you, again, kind of combine them all together, you should be able to get something that's pretty high fidelity. Okay, so at the end of all of this, you have technologies you can choose from, you have approaches you can choose from for being able to sequence the DNA of your mysterious sea creature. And now I want you to think, like, what can you learn? What secrets does your sea creature's genome contain? Maybe there are new pigment proteins. Maybe there's new medicinal compounds or enzymes that make new medicinal compounds encoded in the DNA. Maybe there's instructions for limb regeneration, losing a whole tentacle. That's not something that organisms do lightly. Usually they have a strategy to regrow that lost limb. Maybe there's new bioluminescent proteins or instructions for how to withstand high pressures. Maybe there's something else, but I think only once we sequence will we really know a little bit more about what the potential of this organism might be. Okay, so as you were listening today, I'd like you to kind of reflect back on what would you like to sequence if you had the capacity to sequence something? What are the potential benefits of sequencing an organism's genome? What would you wish for in the next generation of sequencing technologies? I told you a little bit about the advantages and disadvantages of the ones that we already have, but are there any other things that you might wish for a DNA sequencing technology to be capable of? And then if SmidgION became available tomorrow, would you buy one? What would you do with it? I think that's fun to think about. All right.